Yes, well, thank you very much, and thank you for the invitation and for setting up this webinar. What I'll do is I'll give an overview of the overlap between cancer and aging, but without going into a lot of detail about the major potential processes at play, and also touching on some of the issues that then the other speakers will cover in more detail. So, as I'm sure you're aware, cancer incidence increases dramatically with age. Cancer and age-related diseases affect typically older individuals. And as you can see, for example, on the right here, you can see that's a log scale. So it's the same data on the left and the right, but on the right, it's on the log scale. And as you can see, so cancer incidence increases exponentially with age. And this is true for the vast majority of tumor types, although the pace of increase with age can differ between them. There are a few exceptions, however, the most notable of which is testicular cancer, which quite strikingly has an incident peak relatively early in life, around age 30, which is quite surprising, and the mechanisms or the reasons for which are not well understood yet. Having said that, aging clearly affects or is a major risk factor. Age is a major risk factor for cancer, and when you think about aging, I mean, people are familiar with the aging process, but I'll show you in this check centenarians. The interesting thing about aging is that it affects virtually every organ in the body, and it entails a variety of changes from functional, physiological, molecular, cellular changes. And so I like the definition of aging as a progressive deterioration of physiological function accompanied by an increase in vulnerability and mortality with age. Now, while aging is a complex process that affects multiple systems, we don't yet know what drives the process of aging. I mean, what are the causes of aging? There are a number of theories like DNA damage, which I'll talk in a few minutes, the telomeres, which Maria will talk about later, oxidative damage. So there are a number of theories, but none of which have been proven yet. So I would say that the precise cellular molecular mechanisms that drive the aging process are still under debate. Having said that, we have made some dramatic progress in our understanding of aging. I think arguably the most impressive development has been at the level of understanding that aging is a surprisingly plastic process in model systems and can be manipulated by genes and by environmental manipulations and diet in particular. So at the genetic level, I guess the first gene shown to modulate aging was H1, which was identified in the roundworm C. elegans by Tom Johnson and colleagues over 20 years ago. And it is quite dramatic and at the time even surprising how alleles of a single allele can have a dramatic impact on lifespan and on aging. And we now know of hundreds of genes that have a significant impact on the aging process in model systems. So we know of hundreds in invertebrates and in worms, for example, but even in mice, there's about 100 genes that we know of that have a significant impact on aging phenotypes. So we have a number of genes that if you disrupt them, they result in a significant increase in lifespan. So I believe the current record is a growth hormone receptor, which if you knock out in mice, you obtain dwarf animals that live about 50% longer than wild-type controls. And what is interesting as well is that these animals not only live longer, but they live healthier. And I'll mention this in more detail in a little bit, but cancer is one of the diseases that is affected or that is retarded when lifespan is increased in this model. Conversely, there's a number of models also of accelerated aging in which disruption of particular genes result in what is called segmental progeroid syndromes in mice that resemble accelerated aging. A number of these genes are actually related to DNA repair and DNA damage responses. And so, not surprisingly, one of potential areas of overlap between cancer and aging is exactly in terms of DNA damage, because we know that mutations are one of the hallmarks of cancer, and we also know that DNA damage and DNA mutations accumulate with age. And so one of the theories of aging, the DNA damage theory of aging, argues that with age, there's an accumulation of DNA damage which impairs cell function, which results in cell loss, possibly at the level of stem cells as well, and that this is one of the main causes of aging. There is some evidence for it, as I mentioned before, in terms of this segmental proteroid syndromes in mice. There's also one very good example from patients, which is Werner syndrome, which is arguably the most striking proteroid syndrome in people, which is what you see here. As you can see, these patients look older at younger ages than they normally would, and these individuals develop a variety of diseases at early ages, including cancer, although the exact type of cancer that they developed is not the same, or is not exactly the same as in normal individuals. But what is remarkable is that this disease, which results from mutations in a single gene called the Werner syndrome gene, which is involved in, which is helicase exonuclease involved in DNA metabolism, these individuals develop this complex phenotype that appears to resemble accelerated aging. There's also been a number of studies at the level of quantifying DNA mutation, DNA damage with age. I do think this has not been as clear-cut as probably once thought initially. So, for example, what you see here in this slide is the work of Jan Vig and colleagues. And what they found in mice is that you do see an accumulation of DNA mutations in some tissues, but not in others. So there tends to be, at least for this particular assay that they use, there tends to be tissue-specific effects. And the other thing that is important to emphasize is that while we talk about an overlap between cancer and aging at the level of DNA damage, which is certainly important for cancer, and it appears to be important for aging as well, there are also different types of DNA damage, and there's also different type of DNA repair pathways. And without going into great detail, we know from a variety of mouse models, for example, such as the work of Jan Eichmachers in the Netherlands, that different types of damage can have different types of impact on cancer and aging. So, for example, DNA damage that appears to increase the mutational load, that increases in chromosomal aberrations, tend to impact more on cancer, while on the other hand, DNA damage that results in cell cycle arrest or cell death appears to be more important in cancer. So while there is an overlap between the mechanism, at the conceptual level of DNA damage impacting on aging and cancer, when you go into the details, there may be some differences between the specific types of DNA damage and specific types of DNA repair. Now, I mentioned these proteroid mice and accelerated aging. On the other hand, apparently you have these life-extending mutations in mice, for which there's quite a few. I think arguably the most important pathway involved is the growth hormone IGF-1 pathway. IGF-1 stands for insulin growth factor 1. And we now know that there's a variety of mutations in this pathway in which decreased growth hormone IGF-1 signaling extends lifespan and appears to retard aging. So it's not that just the animals are living longer, they are living longer healthier. So their health span is also extended. And so these animals tend to be smaller than controls, and they live longer. And they also appear to be protected from cancer. I mean, that's a broad generalization. When you look at the different mutants, I mean, some of them have a lower cancer incidence, others have different types of tumors, others have a kind of retarded onset of particular cancer types. But by and large, these manipulations or decreased growth hormone IGF-1 signal does have not just extend lifespan, but also protects against cancer. Which is not too surprising because growth hormone and IGF-1 are essentially mitogens that make cell proliferate, so it makes sense that if you decrease their levels, animals are going to be protected from cancer. And conversely, if you overexpress bovine growth hormone, mice appear to age faster, so they're bigger and they appear to age faster. And this has been the work of, amongst others, Andres Bartke. Now, there's also been a few mutations in individuals that in the same pathway. I'll show you one example, which is in this individuals that have deficiency in the growth hormone receptor. As I mentioned earlier, if you knock out growth hormone receptor in mice, the mice live up to 50% longer, and they're also protected from cancer. And so there was a study conducted in Central America amongst a cohort of such individuals. And what was interesting, or the most interesting finding in my opinion, was that individuals, at least in the cohort, they do not die of cancer. So sample size was not too big. There were 30 individuals, but when compared to their relatives that don't have the growth hormone receptor deficiency, for which about 20% die of cancer, it's quite remarkable that none of these individuals dies of cancer. I should say, however, that these individuals are not long-lived per se. They appear to have a high incidence of some other diseases like heart disease. So unlike the mice, the growth hormone receptor knockout mice who live longer and are cancer protected, these individuals at least appear to be cancer protected but do not live longer. Nonetheless, this shows that the same pathways that affect aging in mice and protect against cancer, at least at level of one specific age-related diseases, in this case cancer, they could be relevant for people. Now, when we talk about environmental manipulations of aging and dietary manipulations in particular, there's been quite a lot of work on this as well for the past 50 years, and by far the most widely studied manipulation is caloric restriction. And caloric restriction was discovered by Claude McKay and colleagues at Cornell, following some earlier findings. And what they found is that so normally mice in the lab, they're fed ad libitum. That means they can eat as much they want. But if you restrict the amount of calories that they eat without malnutrition, this has a significant impact on longevity. It increases lifespan up to 50%, which is this plot you see here, which I believe is from Edwin Masoro's lab in San Antonio. So there is a very significant impact of restricting the amount of calories. And again, just like in the mutant mouse, it's not that the animals are living longer, they're also living longer healthier. And a variety of age-related diseases are also ameliorated by caloric restriction, and cancer in particular. And so if I can summarize 50 years of research on caloric restriction in a single slide, I would say we know now that it extends lifespan in most species, but not all of them. There are a couple of exceptions. We know it does have also some negative side effects. So, for example, it appears to be detrimental in fighting infections. There's been more recently work done on rhesus monkeys under caloric restriction. The work is still ongoing because rhesus monkeys live over 40 years. So as you may imagine, this is a very long project. But the results so far suggest that caloric restriction protects against cancer in rhesus monkeys. Whether it will extend lifespan, I think, is still an open question. There are some conflicting results between two different groups in Wisconsin and NIA. I would say that if caloric restriction extends lifespan in rhesus monkeys, it will be far less than what it does in rodents. So if you extrapolate to primates, it appears that caloric restriction does have cancer protecting effects, but in terms of its impact on longevity per se, it's probably less than what we observe in rodents. And so my own view is that caloric restriction isn't likely to be as effective in humans. I mean, I certainly don't think it's going to be as effective in humans as it is in rodents, and the results from rhesus monkeys support that. But particularly given its impact on age-related conditions and cancer in particular, it could be helpful in trying to identify pathways involved and try to identify drug targets that could be used in the clinic, in particular for treatment of age-related diseases like cancer and type 2 diabetes. And there's a number of work focusing on this. And I mean, my own view of caloric restriction is that it involves endocrine mechanisms like growth hormone, insulin, IGF-1, but also there's a complex signaling cascade involving things like PI3K, FOXO, and TOR, and many other players. That is also important. If you can understand this, then it may provide applications in the clinic for age-related diseases. And so what we do know is that there are longevity pathways that can modulate aging, and they do so to a very dramatic extent, and even surprisingly. I think people didn't expect this impact 30 or 40 years ago. And we do know that these pathways also tend to impact on cancer. So the same mechanisms of caloric restriction, for example, and these pathways that retard aging in rodents also have a significant impact on cancer. And conversely, you have DNA damage and DNA repair systems that also probably are involved in aging and are certainly involved in cancer. Although, as I mentioned earlier, they could be slightly different types of DNA damage and different types of DNA repair that are more important for cancer and aging. And so now the last thing I will mention is whether. So we know, for example, that mutations accumulate with age, and we know that that is important for cancer. But there's a number of changes in all of our organs during age. So I think a crucial question as well in terms of the relationship between aging and cancer is whether tissues of all tissues, whether there are more fertile ground for cancer development. And actually, contrary to popular perception or sort of a urban myth, is that so cancer is not less aggressive in the age. I mean, there are some types of cancer that are less aggressive in the age, most notably breast cancer, probably due to genotype differences. But by and large, cancer is not less aggressive in the age. On the contrary, it's actually more aggressive in the age. I mean, there's some very clear examples like ovarian cancer, which is clearly more aggressive in the age, and that is the rule, not the exception. So cancer does tend to be more aggressive in the age. So one question is whether there are processes related to aging or aging deterioration that contributes to this. There's been some work, which I will briefly summarize on this. One potential area of interest is related to senescent cells. These are cells that suffer from irreversible growth arrest, either due to short telomeres or stress. And this is mostly the work of Judy Kempisi's lab. And what they've shown is that senescent cells, which tend to accumulate with age, at least in some tissues, these contribute to create a tissue microenvironment that facilitates cancer development. And they've actually done some elegant experiments in injecting mice with malignant cells with senescent cells and with non-senescent cells. And what they showed was that if you inject mice with malignant cells together with senescent cells, this forms more and larger tumors. Having said that, there's probably several types of processes of aging that contribute to both help cancer development and also hinder cancer development. So on one hand, we know that time exposure is a crucial aspect in cancer. I mean, there's mutations that accumulate. So what you see here in blue are cells accumulating mutations. But on the other hand, you have a variety of aging processes that could also both help and hinder cancer development. So, for example, in terms of helping cancer development, I mentioned senescent cells, but another very important process in aging is inflammation. So we know that inflammation is important in age-related diseases. We know inflammatory levels increase with age, and we know inflammation is also important in at least some types of tumors. So it is certainly conceivable to hypothesize that age-related changes, increases in inflammatory levels, contribute to cancer development in the age. While conversely, you could have even mechanisms that hinder cancer development, and one obvious example would be hormonal levels. So I mentioned earlier how growth hormone and IGF-1, if you reduce growth hormone and IGF-1 signaling in mice, it extends lifespan and retards cancer development. So what we also know is that the levels of these hormones decline with age normally. So it's conceivable to hypothesize again that this actually hinders cancer development in the elderly. So there's a variety of processes, and it will also depend on cancer type and tissue type. So to summarize, we do know that some aging changes may render tissues more prone for cancer development, like senescent cells and inflammation. On the other hand, if you have normal processes that maybe degenerate with age but are important for tumor development, then this changes with age may contribute to cancer resistance. And there's been a variety of studies using xenografts in animals of different ages, where you inject malignant cells in animals of different ages. But these have been providing conflicting results. So sometimes it forms these experiments, you observe more tumors in young, other times in old animals. But what I do think they show is that there's more to how age is a factor in cancer development than merely mutation accumulation. And I think there's a lot of work still to be done in this. The one thing I would emphasize is that there's also a lot of tissue and tumor-specific effects. So it's difficult to generalize how this process, the aging processes, affect cancer. And to finalize, I mean, there's a lot of work still to be done in this area in understanding the overlap between aging and cancer. There's still a lot of open questions. I mean, we still don't fully understand why testicular cancer incidence peaks so early in life. As I just mentioned, I still think there's a lot of work to be done in understanding which tissues are affected, in which way to increase or to be more or less fertile for tumor development, the issue of how hormones and how the immune system changes with age, how this influence cancer development. I think there's been very little study in so far. And lastly, because we know that longevity pathways and dietary manipulations like caloric restriction impact on aging and cancer, certainly one exciting prospect is whether we can employ this knowledge of these pathways and the players involved to ultimately translate these findings to improve people's life and to develop anti-cancer treatments. So thank you very much. And so this is just some information about our lab if you wish to learn more about what we do. Thank you.
Thank you, Dr. Megalish, for a great presentation. We have a question from the audience for you, and I will read the question is, has the research on caloric restriction accounted for the fact that eating a smaller amount of calories would be likely mean this is a lower consumption of carcinogens through the food supply? In short, is it the caloric restriction that is protecting against cancer, or is it the decreased amount of carcinogens that someone would ingest that would decrease the risk of cancer?
So I think there's two issues that you touched there. So the first one is what constitutes a normal diet, in the sense that, okay, so you're doing caloric restriction, but on the other hand, well, maybe the problem is with the control animal that maybe they're eating too much of something that is bad for them. And I think that's been an issue in the field for a very long time. And there's actually an issue with the studies of rhesus monkeys, because some of the differences in the studies that have been published so far relate to how the controls are treated. I mean, what is a normal diet? I think the take-home message in general is that eating a lot of calories will be detrimental, both in terms of lifespan and in terms of cancer. As for the other issue, which is whether in the food intake, whether there's carcinogens, that is something a little bit outside of my expertise, but I wouldn't necessarily think that there are carcinogens in the normal diet of animals in the lab, depending on how you define a carcinogen or a mutagen. So I normally would not think that to be a problem. So my guess is not that it's the food that is causing the cancer, rather there are pathways involved. There's a lot of work on obesity and cancer, and I think Steve will talk a little bit about this as well in his talk. So in terms of the metabolism, and that this in turn impacts on cancer, but not the carcinogens in the food themselves.
Okay, thank you. And we have time for one more question. what percentage of caloric restriction was associated with decreased aging in animal models?
I think there's a variety. If my memory serves me correct, there's a variety of diets that can result in a lifespan increase. I do think that the most striking effects are done with the most severe diets without malnutrition. So I think you can do like something like 40% reduction of calories. I have seen studies along those ways, but there are other variants that can also result in an increase in lifespan. So there's different types of restriction of diets that can have an impact, though I think that the ones that have the most impact are the strictest ones, so to speak.
Thank you so much, Dr. Megalish, for a great presentation and for answering the questions. Again, for additional questions that we are unable to ask you at this point, we will follow up with the participants and forward to your email address the questions to answer later. And now we need to move on, and I'm pleased to introduce our next speaker, Dr. Maria Blasco. She is the director and the head of the Telomeres and Telomeres group at the Spanish National Cancer Research in Madrid, Spain. Dr. Blasco will present the role of the telomeres in health span and longevity. When Dr. Blasco is finished, we'll have time for a few questions as well. Dr. Blasco?
Hello. It's a pleasure for me to give this webinar. So my talk is going to start by saying that one of the highest risk factors for developing cancer is aging, and I think this is true for other diseases as we just heard. So incidence increases with age, and from 40 to 50 years old onwards, increases very dramatically. So this means that aging is one of the causes of disease. This is our view of the problem. While we are young, and there is little cellular aging, also there is less disease, and is aging what causes the disease. So aging would be the cause, and the disease would be the consequence of this aging process. It's very important to understand which is the cause of aging. There are genetic factors and environmental factors, because if we are able to find these common causes of disease, then therapeutic interventions which are aimed to treat disease would be able to delay aging-associated diseases. So in other words, we would be able to increase health span and delay the appearance of diseases. So this is a change in paradigm. You can think that you can develop therapeutic interventions which are different for every particular disease, and this has been the situation until now. Or you can think that there is a common cause for disease, which is the aging process, and you will be able to develop therapeutic interventions which are directed to slow down the aging process, and in this manner being able to prevent or treat many different diseases. So which are these causes of aging? So during the last, I would say, 20 years, there have been strong genetic evidence identify a number of molecular pathways. We heard some of them, which are important to explain why cellular aging happens. And my work has been working in one of them, telomere shortening. And I will describe about our work regarding the role of telomeres in aging and disease. So the telomeres are the ends of eukaryotic chromosomes. They are very important to protect chromosomes from fusion and rearrangements. And we also know that telomeres, however, they do shorten with every cell division. This is a defect which is accumulative with increasing cell divisions. Life would not be actually possible if there would not be a way to compensate for this telomere loss. And in fact, there is an enzyme which is known as telomerase. Telomerase is a pluripotency enzyme, so it has development and is able to recognize the chromosome end and by copying an RNA molecule, which is part of the enzyme, it's able to compensate for this telomere shortening. So telomerase resets telomere length at the blastocyst stage. When we are born, telomerase is silenced in the majority of cell types and tissues, including also some stem cell compartments, and this causes telomere shortening. When telomeres become critically short, this triggers a DNA damage response, leading to cell death or cellular senescence. And we think that this is one of the causes why aging happens. In the case of cancer, however, cancer cells need to reactivate telomerase, and this happens in the majority of human cancers, in order to be able to maintain the telomeres always young and to be able to divide immortally. So this is the interest of studying telomerase in cancer and aging. This is another way to look at the problem. So telomere length decreases with increasing age. This includes the stem cell compartments. When telomeres reach a critically short length, this would be causative of disease. Cancer is an aging-associated disease as well, but in the case of cancer, telomerase have to be reactivated in order to allow the immortal growth of cancer cells. So we know that there are some human diseases which are known as telomere syndromes, which are humans that have mutations in some of the telomerase or telomere components, and they have a premature loss of the regenerative capacity of tissues and premature death by different diseases like aplastic anemia or pulmonary fibrosis, among others. So this indicates that the amount of telomerase that we have are very limiting, and defect in telomerase will lead to a premature loss of the ability of our tissues to regenerate. We also know that not all individuals of the same age have the same telomere length. Actually, telomere length is very heterogeneous, and we know that there is a correlation between having shorter telomeres than normal, being in a lower percentile of telomere length, with a higher risk of developing different diseases. And this brought the possibility that telomere length could be used as a biomarker of aging with prognostic value for many different diseases. Actually, in my group, we have been exploring this possibility, and we have developed a technology, which is a high-throughput technology that is able to measure telomeres in blood samples, so you can determine both the mean telomere length and, what is more important, the abundance of short telomeres, which are the ones that trigger the DNA damage response. So we have validated this technology in a recent collaboration with a group of Michael Snyder in Stanford, University of Stanford. Michael Snyder has a mutation in telomerase, and when we measure average telomere length with our technology, it can be measured by other technologies, we see that Michael Snyder has a normal telomere length when compared with individuals of the same age. However, when we look at the abundance of short telomeres, which, as I said, are the ones that cause disease, we see that Michael Snyder has a very high percentage of critically short telomeres, in this case telomeres below 3 kilobases, and therefore we can correlate the phenotype, which is a mutation in telomerase, with the phenotype, which is having shorter telomeres than normal, with the genotype, which is a mutation in telomerase, and we think this could be valuable for disease prevention. So currently we are exploring many different diseases which have been related to presence of short telomeres, and I will give you an example with familiar breast cancer, and which brings me to the following reasoning, is that we know that genetic testings are important to determine the risk for developing diseases, but they do not reflect on the way of life or the life habits. And telomere length, however, they are both an indication of the inheritance, so you can have a mutation in telomerase, and also of the environment. And there are many different publications showing that the rate at which telomeres shorten with aging depends on life habits, and this is relevant. And we think that the combination of both genetic information, and in this case, telomere length, would give us a more powerful way to predict risk and time of onset of disease. And of course, this could also aid in the development of new treatments to prevent disease. In the case of breast cancer, familial breast cancer, you know that mutations in BRCA1 and BRCA2 give a higher risk of developing breast cancer. And in here at the CNIO, the group of Vietnamese demonstrated that when you combine genetic information with telomere length, this is a more powerful way to predict risk. So the women that have the mutations in BRCA1 and BRCA2 and have shorter telomeres develop breast cancer earlier in life. So it's a factor that helps assessing risks in the case of breast cancer. So one of the questions, of course, is can telomere length also predict lifespan of a given individual? And I would like to mention a couple of studies. In humans, we don't know yet, but in birds, there was a study which showed that, a longitudinal study that showed that telomere length at day 25 after birth was predictive of the longevity of this bird species. And we have done a similar study with mice. So we perform a longitudinal study, so following telomere length in individual mice with time, and see whether any parameter associated to telomeres was able to predict longevity in mice. And the first thing that we observed is that mice shorten their telomeres 100 times faster than humans. And this is interesting because this may explain why, even though mice are born with longer telomeres than humans, they live for much shorter time than humans. So their telomeres also shorten much faster than human telomeres. And what is important is that we found that the rate of increase of short telomeres was predictive of individual mouse longevity in two different strains, a wild-type strain and also a strain which had increased telomerase expression that we knew that were longer lived. They were delay aging. So, of course, knowing that short telomeres can be causative of aging, you can think about possible therapeutic strategies to delay this process. In this review that we published, we propose that in the case of telomere shortening, activation of telomerase in adult tissues, where we know that telomerase is silenced, could be a therapeutic strategy. And I would like to give you some data on that regard that we have published. So on the one hand, inactivation of telomerase by making a deletion of the telomerase gene in mice causes premature aging, very similar phenotypes to these telomere syndromes, even though the studies were done before in mice than in humans. And also, they were useful to validate that telomerase could be a good anti-cancer target because these mice also had less cancer because they didn't have telomerase. When we increased telomerase in adult tissues, using first transgenesis, just making transgenic mice, we saw that only overexpressing telomerase was resulting in less aging, but however, we saw that there was a slightly more cancer and the longevity was not increased due to this increase in cancer. Because, as I said, telomerase is important also for cancer cells to divide. So the way we had to solve this was combining telomerase overexpression with increased expression of tumor suppressor genes, and this resulted in a delay aging and an increase in lifespan, which was very significant. And actually, this was the first demonstration that telomerase can extend lifespan in any organism. And as here you can see that telomerase overexpression in this cancer-resistant context can increase median longevity by 40%. Not only that, but if we look at the health span, so which in a wild-type mouse is around 1 year. After 1 year, mice start developing different diseases, including cancer. In this strain of mice, we were increasing lifespan by threefold, more or less. So the first mice that died much later than in the case of the control mice. To prove that we are actually increasing, we are delaying aging, or in other words, we are increasing youth in these mice. is that when we do different biomarkers of aging, this is a neuromuscular coordination test. You can see that when mice age, they fail the ability to hold onto a bar, which is something that young mice can do very well. And these mice, the triple mice, the mice with the genetic modifications, including telomerase overexpression, when they are chronologically old, they are able to do the test as well as young mice. So this proves that with a single intervention, in this case, telomerase overexpression, we were able to increase to delay aging. So, because we cannot do human transgenics, we have been also exploring the possibility of developing therapeutic strategies to increase telomerase in adult tissues, in humans, and this is a study that we did in mice. So here we are assuming that mice are aging like humans because they have a deficiency in telomerase, among other things, among other mechanisms. And if we are able to put back telomerase in adult tissues, we will be able to delay aging and delay disease, which shows the adeno-associated viruses, because these are the state-of-the-art vectors that are being used right now in many different clinical trials for human genetic diseases, deficiencies. And the experiment was to treat a single time middle-aged mice or all mice with telomerase or with GFP as a control vector. And you can see here how in the lung, when there is no telomerase activity, of course, in a 2-year-old lung, as you can see here, but in the lungs of mice treated a single time with telomerase, we were able to reactivate telomerase. And this was also interesting because I mentioned that there are telomere syndromes. One of the diseases is actually pulmonary fibrosis, and this could be a way to reactivate telomerase in the lung of these patients. So we see the same as we saw with the transgenic mice, that when we treated with telomerase, we could see an improvement in many different biomarkers of aging. This is again the tightrope success test that I showed you before. The mice treated with telomerase, they perform better in this test. And what was important for us is the longevity. So with a single treatment of telomerase in the middle age or in the old group, we could see an increase both in the median and in the maximum lifespan. Both were increased, even in very old mice, in mice that were 2-year-old. And this was in the absence of more cancer. So the incidence of tumors was the same in the control group or in the telomerase treated. So to finish, I would like to mention something, and this is, I forgot, this is the summary of the experiment. So we are treating mice with telomerase. This is sufficient. I didn't show you, but we showed that. This is sufficient to delay the rate of telomere shortening and, again, to delay aging and increase longevity. So I wanted to finish by mentioning the impact of life habits, in this case calorie restriction, on telomere maintenance. And for that, what we did is a longitudinal study in mice, where we are going to follow the same mice over time during the whole lifespan in 2 groups, mice that are 4 groups, mice that are calories restricted or they are having a normal diet. The normal diet is also controlled in terms of calories, so the mice are not eating ad libitum, they are eating a controlled number of calories. And we are using 2 different strains, wild-type mice or mice that overexpress telomerase, which have slightly different longevities. This is just to show that the protocol is working, and the mice that are under calorie restriction, which are in light blue, they have a lower weight, as you might expect. They have lower fat content. Metabolic parameters are also improved, like glucose tolerance and insulin levels, etc. And we see a tendency to see bigger effects of calorie restriction when we use the calorie restriction in the telomerase overexpressing mice, and I will mention that also later. We see also a lower decrease of bone density in the mice that are calorie restricted, as well as an improvement in this neuromuscular coordination test that I showed you before. Mice that are calorie restricted from both genotypes, they show a better performance at 1 year here after diet. So what happens with telomeres? So what we see is that telomeres are also better maintained in the mice that are under calorie restriction. You can see here on the top telomere length decreasing with age in wild-type mice, like in humans. Mice also shorten their telomeres, as I said before, and a corresponding increase in the percentage of short telomeres in mice. And how this trend is similar in the two strains, in wild-type and telomere transgenics. However, in the mice that are calorie restricted, we see that after initial decrease in telomere length, then telomeres are maintained. And also there is not an increase of short telomeres. So this results in having longer telomeres with time in the mice that are calorie restricted compared to the control mice. We can see individually every mouse and see how it behaves. And we observed that in the case of wild-type mice, 100% of the mice that were under the control diet, they show telomere shortening. So like in the green lines here in the graph. However, 20% of the mice, wild-type mice that were calorie restricted, they were actually able to elongate telomeres over time, which is something that never happens in wild-type mice under controlled diet. This ability to elongate telomeres was also seen in some of the telomerase transgenic mice, as you may expect, because they are overexpressing telomerase. Also, when we look at the percentage of short telomeres, we saw that the wild-type mice under calorie restriction, but not the ones that were under the control diet, were able to decrease the abundance of short telomeres over time, which are here shown in red lines, which is again indicating that calorie restriction has a beneficial effect on telomere maintenance. We have looked also in different tissues. These are not longitudinal studies, but at the end point, we measure telomeres in the kidney, the lung, the muscles, and in all cases, we see an improved telomere length or better telomere maintenance in the calorie-restricted mice. We have also looked in the bone marrow, where we can actually look at metaphases and look not only at telomere length, but also chromosomal instability associated with telomere dysfunction. And we see that telomeres are better maintained in the calorie-restricted mice. And also there is less chromosomes which lack telomeric signals, or less chromosomes which have a type of aberration that we call multitelomeric signals, which are an indication of replicative stress at the telomere. So suggesting that calorie restriction may be decreasing replicative problems at telomeres, and that's why also they have a beneficial effect on telomere length. And just to mention finally that we were able to see also a synergistic effect of dietary restriction with TERT overexpression, with telomerase overexpression. So as I mentioned before, when we overexpress telomerase alone, there is more cancer. But if we combine this with calorie restriction, we are canceling the pro-tumorigenic effect of telomerase, like we did before with combining telomerase with tumor suppressors. So in this sense, calorie restriction is acting as a tumor suppressor mechanism. And when combined with telomerase, we cancel the effect of telomerase in increasing cancer. And as a consequence of this, we think, and also as a consequence of having longer telomeres, we see a significant synergism also in increasing longevity, in the combination between telomerase overexpression and calorie restriction. And the conclusion of my talk will be that we think that there are common causes for many different diseases. One of these common causes is telomere loss, which can be influenced both by genetic and by environmental factors, and that if we are able to delay this telomere loss in this case by an intervention, which is telomerase activation in adult tissues, then we're able to extend the time of life free of disease and also increase longevity. And my last slide is the people of the lab that have done the work that I mentioned to you here. And finally, the acknowledgements of the funding agencies. Thank you very much.
Thank you so much, Dr. Blasco. I have few questions for you from the audience. And the first question is, are there known variants in the human population of individuals who lose slower or who do not lose telomere activity? And if so, what is the phenotype?
Yeah, well, this is a very interesting question. What we know so far is that there are telomere length is heterogeneous, and not all individuals shorten telomeres at the same pace. We know some of the genetic causes of that, like being mutant for telomerase or some of the telomere binding proteins, but we don't know whether individuals that have longer telomeres, they have some, we don't know which genetic alterations may have individuals that have much longer telomeres than normal. We know what causes shorter telomeres than normal, but not the other way around. But I think it's a very interesting question.
Yeah, yes, I agree. It would be nice to know that as well. We have one other question is related to the stem cell dysfunction, and you said that telomerase deficiency would result in stem cell dysfunction, but how would the percentage and the status of stem cells is different in young animals or young people versus old animals or old people?
Yeah, we have done the study precisely in mice. So we have a study in particular the telomerase deficient mice, which is an artificial model where we induce dramatic telomere shortening. And we saw that when mice have critically short telomeres, actually we saw that there are more label retaining cells, which is the technique that we use to identify stem cells. In particular, we did studies in the skin. We saw that there were more putative stem cells in the skin when telomeres were short, but these stem cells were not able to react to stimuli, like when we treated with agents that are known to mobilize stem cells. The stem cells that had critically short telomeres were not responding to the mitogens in this case, and they were not mobilizing, and they were not regenerating the skin or the hair. So what happens, we think, when stem cells have critically short telomeres is that they are there, but they are not able to actually mobilize and regenerate tissues.
And this would be the last question. How is telomere length and cancer rate in young people with the aging syndrome compared with normal people on the same age?
You mean with the telomere syndromes? Because there are other aging syndromes which are not related to telomere shortening. But in the case of the telomere shortening syndromes, in the telomere syndromes, they have an increased cancer as well. So because, as I mentioned with the breast cancer study, so aging is also, and telomere shortening seems also to be increasing the risk of cancer. And individuals that have faster rates of telomere shortening because they are mutants for some of the telomere binding proteins or telomerase, they also seem to have an increased risk of cancer.
Thank you so much, Dr. Blasco, for a great presentation and for answering the questions. And we will follow up with you with additional questions that we have, and the time is not permitting now to answer all of them. And we're going to move on to our third and final speaker, Dr. Steve Hursting. Dr. Hursting is a professor, McKean-Lab endowed chair of nutritional, molecular, and cellular sciences in the Department of Nutritional Sciences at University of Texas. And Dr. Hursting will discuss the role of nutrition at the intersection of aging and cancer. At the end, we'll have time for a few more questions. Dr. Hursting?
Thank you very much, Gabriela. And hello to everyone from beautiful Austin, Texas. It's an honor to join you today and talk about nutrition, cancer, and aging. But I'd like to cover some of the links between diet and aging and the cancer process, both in humans and animals. And I'll focus on obesity, which, as we know, has emerged as a major risk factor for cancer, many types of cancer, and is also really contributing to some accelerated aging, and we'll talk about that. We'll also particularly focus on lessons from aging research in terms of molecular targets and strategies to break some of the links between obesity and cancer. We've heard about some of these already in today's webinar. We'll focus on IGF-1, insulin-like growth factor 1, and its downstream signals, adipokines and inflammatory signals. There are some emerging targets that we've heard mentioned already. Certainly telomerase, we just heard Priya, but sirtuins and their pathway also seem to be coming into the fore. And we'll wrap up with a discussion about possible diet and or pharmacologic strategies to break these obesity cancer links. So that's what we'll cover. Now, if you pick up a newspaper or magazine, almost always there's a reference to this obesity epidemic that's occurring in the United States. And certainly we're not alone. There are an estimated 700 million obese adults in the world, many countries affected by this. In the U.S., over the last 30 years, we've seen our obesity prevalence more than double. And about 7 out of 10 adults are currently obese. It's determined by a BMI greater than 25. About half of those, about 36% currently of our U.S. adults are in the obese category, BMI greater than 30. And there is an age effect here. There's even a higher rate. Approximately 42% of our U.S. adults in the 65 to 74-year category are obese. This certainly has a major impact on a number of diseases related to aging. So this obese phenotype we know is associated with alterations in metabolism. In fact, in more than half of the individuals with this phenotype, we actually refer to them as a metabolic syndrome, and they're characterized by insulin resistance and hyperglycemia, dyslipidemia, waist circumference, the classic factors. There are also alterations in inflammation. Obesity really is a pro-inflammatory state, and we see increased cytokines and chemokines in circulation in these individuals. There are vascular perturbations linked to increased factors such as vascular endothelial growth factor and plasminogen activator inhibitor 1, but these can impact vascular health and neovascularization as occurs with cancer. There are altered adipokines. Particularly, we see increases in leptin and decreases in adiponectin. These factors produced by fat cells that have a number of biologic effects. And as we've heard already, certainly IGF-1, insulin growth factor 1, is elevated, particularly the bioavailable form of IGF-1 in its relationship with a number of binding proteins that impact bioavailability. So there's a whole spectrum of really metabolic perturbations associated with this phenotype. And we know, a number of chronic diseases. For many years, we've known that, for example, diabetes and heart disease and hypertension have been linked to this phenotype. Really, it's just been the past decade, maybe a little longer than that now, where we've had the appreciation of how major a risk factor obesity is for a number of types of cancer. And recent estimates, particularly, I'll alert you to a paper by Graham Colditz last year, did an analysis, and an estimate of roughly 25% or nearly 150,000 cancer deaths per year are caused by this overweight obesity state. And so this is a major risk factor, something we've got to get a handle on in terms of breaking this link between obesity and cancer. Now, energy balance is certainly sort of the driver here. We've got a nation increasingly in a positive energy balance state, and I think it's worth looking at the sort of energy balance equation and how it relates to aging and cancer. On the energy in side, we're really referring to amount of calories consumed. The type of calorie can also have an impact, whether it's a carbohydrate or a fat calorie, or even within carbohydrates, the type of calories, for example. Pattern, if the calories are spread over the course of a day versus maybe 3 large, sort of the traditional American 3 square meal a day kind of approach, that can have an impact as well. On the energy outside, really the only modulatable aspect of this side of the equation is physical activity. It's very difficult to modulate metabolic rate, thermoregulatory mechanisms, growth or storage mechanisms. So really physical activity, increased exercise is certainly a target here and is really the only modulatable aspect. Now, in terms of the connections between energy balance, aging, and cancer, as we heard already, the best studied aspect really is the energy inside, and particularly calorie intake, calorie restriction. There's a large literature on. And again, just to define what we're talking about with calorie restriction, it's really a reduced calorie intake and typically a 20 to 40% reduction relative to an ad libitum fed control. Without malnutrition. So pains are taken to ensure adequate nutrition, adequate vitamin, mineral, amino acid, fatty acid, nutriture is there. It's really a reduction in energy is what we're talking about. And it's the best studied of this in terms of its connection both to aging and as already has been mentioned, calorie restriction extends lifespan in multiple species. Here I've just taken example where a 20% reduction was reported in these studies. And you see across organisms, yeast, the worm, C. elegans, fruit fly, the bull and doily spider, even dogs and cows have been looked at in this regard. And a 20% reduction in calories results in at least a 20 to perhaps even 40% reduction in longevity in some of these cases. So, a consistent, strong effect of calorie restriction in terms of both median and maximal lifespan has been seen in a number of organisms. Best studied certainly has been the rodent, and I was struck by this paper. I started my graduate work in 1987, and this paper by Rick Weindrich in Journal of Nutrition had just come out in '86, showing that compared to a non-calorie restricted group, 25, 55, or 65% calorie restriction leads to an extension in lifespan. And Rick Weindrich kind of equated that to human lifespan equivalents in years. And so you see a median lifespan in the mice of about 29 months in the non-calorie restricted. With a 25% reduction in calories, you see an extension to a median to something like 100 or so years. In terms of human equivalents, in about 39 months or so in terms of the mouse lifespan, and increasing with 55% even a more extended lifespan. This does show that this can be sort of maximized, and in fact you can run into malnutrition much above 40%. So most of the studies are done in that 20 to 40% range. But a clear effect, and this has been consistently shown, a life extension effect of calorie restriction in a number of rodent species. Now, how about cancer? And certainly part of this life extension is a reduction in tumor development. And Pedro even mentioned earlier that this seems to be occurring in monkeys as well. But in the rodent systems, a number of I'll just show you spontaneous tumor development in a number of experimental model systems, mammary, liver, leukemia, and so on. Number of mouse and rat strains used for this, and a variety of degrees of calorie restriction. I've limited to 20 to 40%. But what it sees is a very consistent and strong effect in terms of the ratio of incidence model systems, comparing the ad lib group to the calorie restricted group. So that ratio, think of that as something of a relative risk. And it's on the order of anywhere between 2 to 5, all the way up getting close to 50-fold increases. And what you see is always positive, and it's always, in many cases, quite strong, this effect of calorie restriction in reducing the development of cancer in these models. This is spontaneous tumors. There's a huge literature on chemically induced tumor model systems. Same story, very strong, very consistent effect on tumor development in response to calorie restriction. Now, a number of mechanisms have been proposed for how this might work. I've taken this from an article from Congo and Fontana, that shows that a decreased calorie intake. This is really sort of a checklist of some of the known adaptations that occur with calorie restriction. We see it at the metabolic level. We've already mentioned some of these: insulin, like growth factor 1 insulin, sex steroid and hormones and cortisol, number of oxidative stress markers, inflammation, adiponectin we mentioned, and thyroid hormones. So there are clear metabolic adaptations. At the molecular level, what's striking is particularly, we've already mentioned the mTOR pathway. A number of components of this IGF insulin signaling pathway through PI3 kinase down through mTOR is involved. Number of those components. Also, oxidative stress components mediated by the antioxidant regulator NRF2. Sirtuin pathway we'll mention in a minute, but that also is emerging. So a number of molecular alterations. And in terms of cellular adaptations, we see generally a decrease in cell proliferation. Apoptosis generally increased. The process of autophagy is increased. We see DNA repair and carcinogen detoxification processes improved in this low-calorie environment. Oxidative damage is decreased. Genomic stability, possibly involving SIRT1 regulatory mechanisms there. And immunosurveillance generally improved in terms of anti-cancer immunosurveillance. And the net result here, as we said, is a consistent story of an increase in median and maximal lifespan. For many years, this was really the only experimental modality to increase median and maximal lifespan. And also, we know a decrease in cancer risk and progression. So these two linked processes are occurring and responding to calorie restriction. Now, we can gain some clues from several long-lived mutant organisms. We've already seen that calorie restriction affects both the lifespan and in what higher organisms, cancer in these organisms. One clue that we've gotten from some of these long-lived mutant organisms again relates to this insulin-like growth factor 1 TOR pathway. So we see in C. elegans, there's a mutant called DAF2 that is really an alteration in this highly conserved pathway, IGF-1 pathway, all the way down to C. elegans. Mutations in that pathway can lead to life extension in C. elegans. Also in the fruit fly, there's a Chico mutant that is an IGF-1 receptor mutation. Again, a life extension in lifespan in occurrence with that IGF-1 mutation. And in mice, we can actually manipulate genetically the components of this pathway. And for example, here's an IGF receptor heterozygote that has an extended lifespan relative to control. So some evidence that indeed this pathway is important in life span and the aging process is really quite clear. And again, in rodents, we can manipulate these pathways, and one approach that we've taken is to knock out IGF-1 in the liver. Most of our insulin-like growth factor 1 in circulation, it comes from the liver, about 80% or so. And so we used a Cree-Lock approach to actually knock it out and try to mimic what we kind of are seeing with calorie restriction, which is a reduction in IGF-1 levels in response to the diet. So this is a genetic approach to try to mimic that. And indeed, these liver IGF deficients or LID mice have about a 55-60% reduction in circulating IGF-1. And as already been mentioned, there are examples in humans of individuals with alterations in these pathways. So here's an example from the Laron syndrome. A group of Ecuadorians, or sort of a cluster in Ecuador of these Laron syndrome individuals. And as I think has already been mentioned, some of these dwarf syndrome individuals have very low IGF-1, they have very low inflammatory cytokines, they tend to have a trend toward longevity, and virtually no cancer or diabetes in these individuals. And so there seems to be some clear evidence that this pathway is important. In terms of cancer, we've looked at this in a number of model systems, and one where we see a particularly response to calorie restriction is we've used this MMTV, mouse mammary tumor virus-induced Wnt1 transgenic mouse model, and it's a nice mammary cancer model. It's a basal-like model of mammary cancer, and we can actually transplant these tumors into the fat pad of a mouse. In this case, a wild-type mouse, and we see tumor volume as a function of their time after tumor cell injection. And about 6 weeks into the study, the tumors really start to form in the wild-type mice and really take off by about the second month and grow rapidly and kill the animal before 75 days. In contrast, the low IGF-1 group, this LID, liver IGF deficient mouse, never really gets going in terms of tumor growth. We see just barely detectable tumors out at the time when the wild-type tumors are really at a point where we have to terminate the study. So a reduction in tumor development in response to low IGF-1 is clear, and it correlates with what we see in the calorie-restricted state, where about a 50% reduction in IGF-1 is associated with reduced tumor development in a number of models. We've seen this effect as well in multiple models. This is mammary, multiple mammary models, multiple pancreatic cancer models, prostate models, colon. A very similar story, reduced tumor growth and tumor development in the IGF-deficient mice. Now, one thing we were a bit surprised about is the connection between the IGF-1 and inflammation. And we mentioned inflammation as a part of the metabolic perturbation that occurs with obesity and is also associated with the aging process. Every time we manipulate IGF-1, either through diet, through pharmacologic approaches, we see a reduction in circulating cytokines. And here's a list of a panel of cytokines. And in every case, the LID mice, these IGF-deficient mice, are lower than their wild-type parts. And this is true also in calorie restriction. Every cytokine we look at is lower in the restricted mouse than in their ad libitum-fed counterparts. So there seems to be a connection between these pathways that at least we didn't appreciate, and we'll talk more about that as we go. But these interacting pathways, I think, are quite important. Now, we know that IGF-1, insulin, are examples of receptor tyrosine kinases, and so they signal through a pathway, and this TOR pathway, target of rapamycin, that's been mentioned. In the case of the mouse, or other mammals, it's a mammalian TOR, mTOR. That seems to be a critical kind of target, critical collection point for a number of these signals that perhaps gives us an opportunity to intervene. And in this case, we were curious whether IGF-1 and its downstream signal through TOR may be a critical piece to this. And so a grad student in the lab, did a nice experiment where she compared, again, using this WIT1 model, control mice, a 30% calorie-restricted group of mice, and replicated what we saw before, which is a reduction in tumor growth in response to the calorie restriction. But then Letitia took another step. She added IGF-1 back to these animals, because we know there's about a 50% reduction in IGF-1 in those CR mice. So these mice are still restricted, but she exogenously added IGF-1 back in, and she rescued much of the tumor growth in that restricted group simply by adding IGF-1. She also repeated the experiment, but in this time she basically locked mTOR on in the cells that she injected. So she compared a wild-type mTOR construct that she introduced into the cells. So they had normal mTOR, and again responded to calorie restriction, as we saw here, with a reduced tumor development in response to the CR. And mTOR was normal, and indeed she saw calorie restriction reducing the activity levels of mTOR. In the group where she locked it on, so she did a constitutively activated, basically a myristoylated mTOR construct, she lost the calorie restriction effect. There was no longer an anti-cancer effect. Even though the animals were restricted, all the hormones changed, etc. They couldn't signal through this mTOR to reduce the growth of the tumor. So further evidence that this pathway is important, and indeed we've seen this in a number of models. As I mentioned, insulin receptor, IGF-1 receptor are examples of receptor tyrosine kinases. They signal through a cascade of enzymes, kinases, with mTOR as sort of the collection point and regulator of this path. And it's a key regulator of both cell proliferation as well as protein translation machinery. So it makes sense that this is what we refer to as an energy sensing or nutrient sensing pathway because these processes are tied in. You don't want to turn on a lot of protein machinery if there's not adequate substrate. So this senses how much energy is in the system and allows this process of protein translation to proceed. We have seen in both in normal and tumor tissue and multiple tumor tissue types, skin, liver, prostate, colon, pancreas, and mammary, that in the obese state, when we have animals fed with a diet-induced obese diet, that every step of this pathway is activated. All these kinases are phosphorylated and with increased phosphorylation. So in this case, activated mTOR signaling. In contrast, every step of the pathway is decreased. We see decreased phosphorylation in all these kinases with calorie restriction. And interestingly, when we compare the liver IGF deficient mice, these live mice, which are not restricted, and they're not particularly lean. They're short, because IGF-1 is low in these animals, but they're not particularly lean. But their IGF-1 levels are low, and they mimic the calorie-restricted animals each step of the way. So it shows that maybe it's not so much the diet, the body size, but the metabolic state that's driving some of these signals. And really, we think mTOR becoming a major target. To kind of further prove that principle that mTOR is involved, and certainly there's evidence in terms of the aging world that it is. I'll cite this paper from Harrison and Nature from a couple of years ago, where they fed in an aging study. They started rapamycin, which is a direct mTOR inhibitor, at 600 days of age. So the animals were sort of eating and living normally, and then at 600 days, rapamycin was introduced into their diet. And what they saw was indeed an extension of lifespan. Here are the males, here are the females. So a separation and extension of lifespan in response to the mTOR inhibitor. So further evidence that this is indeed, in fact, in the aging world, they claim now that in addition to calorie restriction, they now have a second experimental mode to increase median and maximal lifespan. Although really, I think most of the effect is primarily on median lifespan, but still some life-extending effects and so we were curious if mTOR inhibitors, a pharmacologic approach, would have an effect on cancer development as well, and particularly could we offset the effects of obesity. And so Rebecca Deane, grad student lab, did this experiment comparing tumor weight in the lean, calorie-restricted animals, the control, really the overweight control animals, and obese animals that were on a diet-induced obese diet. So the blue bars are just kind of what we've been seeing before. This is the placebo, where the lean, the restricted animals had minimal tumor growth. The intermediate controls had intermediate tumor growth. The obese, rapid tumor growth. If you add RAD-01 or Afinitor, which is an FDA-approved mTOR inhibitor, even in the lean group, there's room for improvement. It basically took the restricted animals and further decreased the tumor development really to a non-detectable level. Most of those tumors were barely palpable. In the controls, it took the kind of intermediate tumor growth down to a calorie-restricted level, even though the animals were still eating a controlled diet, by simply blocking mTOR. So we were able to disconnect some of that effect. And I think most importantly, in the obese group, the addition of the mTOR inhibitor really reversed many of the effects of obesity. So at 10 mg per kg dose, the tumor growth was comparable to the control. Even though the animals were still obese, many of the metabolic perturbations still intact. The only thing we blocked was the mTOR signal. And a little higher dose, and this gets to an issue that we're working hard on right now as well, is this obesity effect on drug resistance. It often takes more drug to have the same effect in the obese state, and that's true even with the Afinitor. About 15 mg per kg, we were able to take the obese mice and really turn them into, in terms of tumor development, the level of a calorie-restricted animal simply by blocking a single target here, mTOR. And so I think that's encouraging that we can indeed develop and identify targets, develop strategies to disconnect some of this obese cancer connection. Here's kind of where we are in our thinking. The calorie restriction through these metabolic alterations, really kind of tuning down the insulin, IGF-1, improving leptin-adiponectin ratio, decreasing cytokines, affecting these vascular factors in a positive way, through their growth factor signaling and inflammatory processes that are related to these factors. And in terms of growth factor, we think this pathway particularly is promising, this mTOR pathway. In terms of inflammation, it really appears that NF-kappa B, the master regulator of a number of cytokines, seems to be quite energy responsive. And COX-2, cyclooxygenase 2, which is a key enzyme involved in prostaglandin metabolism. These two seem to be particular targets in this realm. And that certainly these are contributing to the anti-cancer and anti-aging effects of calorie restriction. Now, there's a big white box in the middle, and that's where I think the future directions go. We're seeing preclinical evidence emerging. We already heard about telomerase from Maria, and so there's a number of factors that I think sirtuins have been a hot topic the last few years, and I'll talk more about that. Certainly, there's a decrease in reactive oxygen species, oxidative stress with calorie restriction. That can certainly contribute. I think genome stability, we heard about epigenetics, telomerase contribute here, and really I think understudied still is the role of immune function and how that can play a role in this connection. So these are, I think, some of the emerging topics. I'll mention just really kind of sirtuin and then wrap up. But in a way, it's kind of a second clue from longitudinal organisms. We know in C. elegans, SIR2 is the human analog of, or the human analog of SIR1 that we're talking about is really a homolog of SIR2 in lower organisms. And what has been shown by particularly Leonard Garante's group and others is that genetic alteration in the SIR2 gene can impact lifespan. So when they introduce an extra SIR2 and they see extended lifespan, when they knock out, mutate the SIR2, they see a trend towards decreased lifespan. And indeed in mice, the SIR1, in these engineered mice with SIR1 overexpression, they're smaller, they have lower body fat, lower insulin, glucose, insulin sensitivity, higher metabolic rates. They really mimic calorie restriction in that regard. And so the story with cancer is just emerging. They're just beginning to really characterize that. But I think this is a promising additional target besides TOR that bears further work. There is a link between CR and longevity that's been beginning to be well established. The lifespan is not extended by calorie restriction in mice that lack the SIR2. Not mice, these are yeast actually, these studies. That lack SIR2. There are SIR2 activating compounds, one of which is resveratrol, the compound, the phytoalexin in red wine, that's gotten some claim from the French paradox. I think the survival of humans lifespan some data emerging possibly in mice as well, and it appears to do it by mimicking calorie restriction. And how, again, from the Garante group, how they think this is working. In calorie excess, like in the obese state, NAD is really probably driving glycolysis. Most of it is shunted in that pathway. And with calorie restriction, reduction in calories, the shunt rate, so NAD really is pushed towards the SIR2 pathway, which is a sort of chromosome silencing pathway through NAD acetylation processes, and that's been linked to increased longevity. So this, I think, is an emerging target and an interesting pathway. And as I said, there are bioactive food components that are known to hit this pathway, including resveratrol, that bear further study. And so that's indicated here. And as mentioning bioactive food components, I'll close here. But there are a number of bioactive food components in fruits and vegetables and other foodstuffs that can, indeed, in preclinical studies, have been shown to influence aging and cancer processes. So, we mentioned we heard about the telomerase story. SIRT1, we just talked about. MicroRNAs also appear to be responsive to a number of bioactive components, and this can certainly impact chromosome stability. There are a number of growth signals, survival signals, mTOR, which we've talked about, MAP kinase. There are bioactive food components. Acid we found to be quite a nice mTOR inhibitor. That's curcumin, the component of curry, can also impact mTOR signaling to an extent. There are bioactive food components known to affect cell cycle machinery, P53 and BCL2, for example, in terms of cell cycle and apoptotic mechanisms. NRF2 has been mentioned, and that clearly is a diet-responsive oxidative stress regulator. Inflammation, again, the curry compound, curcumin, and a number of compounds. Control can also hit COX-2. So there's, I think, emerging evidence that these are candidates. There may be ways to target some of these aging and cancer processes through an approach with body. Some even can mimic the hormonal signals, including insulin and IGF-1 and steroid nicotinisteine that can actually play a role in terms of phytoestrogen activity. So I think there's some real possibilities here to manipulate the system. And in fact, a number of bioactive food components have been established with anti-aging and anti-cancer effects. Again, these are really limited preclinical studies. There's not much bringing this to human, and that's really where I think the focus needs to be. If we can translate, not only as João Pedro mentioned, the translation of this is and particularly coming up with ways. I'd say the biggest challenge of calorie restriction research is no one wants to do it. It's difficult to maintain a low-calorie diet, I think our evidence would say you don't have to go very low to see positive effects. But still, if we can complement by some targeting of these key mechanisms with bioactive food components in addition to low-calorie diets and increased physical activity, I think we can really have an impact on these processes. So I'll stop there, and happy to answer any questions. Here are my collaborators and Sonia. And thank you very much for joining us today, and again, look forward to your questions.
Thank you so much, Dr. Hurting, for such a great presentation. And indeed, we have so many questions for you. I will combine few questions, and then I will forward the rest to you.
Okay.
So the first question is any age recommended to start caloric restriction? So would any age be better than other when we should be starting caloric restriction?
That's a good question. So I think just in terms of practicality and sort of reality, with our grants kind of getting smaller and smaller, we generally study calorie restriction starting at a very young age, and that is we see very strong effects when we do these calorie restriction studies starting at as early in life as six weeks or so. Strong effects. Now that we have studied and others have as well. What happens when you start calorie restriction later in life. And like that rapamycin study I mentioned where they started really about halfway through the lifespan, they began the treatment. We also see protective effects of calorie restriction really almost all the way through the lifespan. And certainly the greatest effect is early. So the earlier the better. But the encouraging thing is it's never too late. And again, equating this to something a 10 or 15% weight loss, if there's some excess weight, we know can have tremendous impacts, no matter what the age, on metabolic alterations, diabetes risk, and so on. We've known that for years, and I think our data would suggest that that's true also in terms of cancer risk. The data, we have some human data that we've been connecting with our animal work with Carol Fabian, a biologist at Kansas. And our data there suggests that in obese individuals, you do have to get the level of restriction has to be sufficient to sort of reverse some of these metabolic perturbations. And so in our data, we really didn't see much effect in terms of some of these cancer-related markers until we had a 10% weight loss in obese women. And so I would say probably the later you start, perhaps the more aggressive one has to be to reverse some of these effects. But to answer the question, I think the good news is that it's never too late in targeting some of these perturbations.
Oh, I'm so happy I still have time. But the next question would be, if someone would start a caloric restriction, like in 5 years after that, should that person decrease even more, or you started at whatever age, and how would you define the caloric restrictions throughout the life?
Yeah, it's a bit of a experimental phenomenon, isn't it? That we kind of package this thing. I would say the key thing in my mind is really preventing what's so characteristic. I mean, the average American's gaining almost 2 pounds a year is our rate of increase here. So preventing that, really maintaining sort of a good healthy weight, and I would say that the key thing within that is a good healthy metabolic state, and preventing the sort of the insulin resistance and inflammation and other components that we've talked about is the key thing. And so whatever it takes to do that, I certainly am not the same weight I was when I was 22 years old. But I'm working on hanging as close to that as I can, and I think that's probably the key. It probably has less to do with the extent of restriction and more to do with sort of maintaining a healthy body size, and probably more important, I mean, that's a good marker, I think, of metabolic state. We all know that there are individuals that there are probably obese individuals with a more healthy metabolic state, and their risk seems to be, for cancer anyway, seems to be lower, and there are individuals that look lean but have a metabolic alteration, maybe some central adiposity that they look like they're lean, but they're picking up some of these inflammatory and insulin-related sort of alterations, and that's an unhealthy state in terms of cancer. So I think that's the key thing, is keeping our metabolic state sort of at that kind of healthy, low risk range. I think that's probably the translational aspect to think about.
So are you saying for people with normal weight, should we recommend, would they follow caloric restriction if they are in normal range for the weight?
Well, and that relates back, I think Joe Pedro mentioned this earlier, that there are a couple of rhesus monkey studies, one where the control group was really pretty heavy, because their control diet was rather enriched with the energy-dense sort of control diet. Their calorie restriction worked very well, really effective at extending lifespan and reducing cancer and diabetes and other things. In the Madison paper that came out about a year ago, their controls were actually on a fairly healthy diet, and so they really weren't obese. And while you saw some of the anti-cancer effects occurring with the calorie-restricted animals, they really didn't pick up a significant lifespan extension. And so I guess my translation of that is that if you're at a healthy weight, good kind of BMI of 22 or 23, wherever you are in that healthy range, you are restricting. I mean, you may be exercising a lot too, but you're probably doing the things that one needs to do to maintain. And it's, I think, an open question whether additional restriction on top of that, what benefit that would have. I mean, there probably are benefits, but you're probably getting most of it because, in a sense, at least relative to our ad libitum fed control human population that we have here generally in the States, that person is probably in a sense at a restricted level, and probably probably pretty well protected. So that'd be my interpretation of that. Whether having if you go from a normal weight to sort of to restrict further into a BMI below say 20, there's the sort of helpfulness of that, and we do see a bit of a U-shaped curve with weight and BMI that one wonders about. Is it wise to go much below that? So that would be my response. I think if someone's already at a good, healthy, BMI of 21, I'm not sure I would necessarily recommend further restriction for those individuals.
Thank you so much, Dr. Stingrid, again for the great presentation and for answering the questions. We will have a few more questions that we will send to you via email because this is all we have time today. And just to summarize, I understand that there are some common pathways of aging and cancer, and we presented today part of them. It seems like the best approach is caloric restriction that will delay aging and may help preventing cancer as well. The caloric restriction should be done as early as possible from what you're saying. And obviously everyone is different. We have a genetic variation. We have different health status. We have the microbiome that has to be part of the equation, and medical history, environmental exposure. So it seems like we cannot make recommendations for the general population other than recommending maintaining an optimal weight, active lifestyle, healthy diet, which we keep saying all the time. And I would like also to thank you again to Dr. João Pedro de Magalhães, Dr. Maria Blasco, Dr. Steve Hursting for their great presentation, and thank you, dear, to our audience for participating today. I would like to thank you, Dr. Harold Seifried, our acting chief, and the rest of the planning and steering committee and the Office of Communications and Education for helping putting this webinar together. And if you have additional questions, please send it to us. My name is Gabriela Riscuta, and you see my email, gabriela.riscuta@nih.gov. Again, to remind you, this webinar is being archived and will be available through our website in about 2 weeks. Also, you will receive an email with information to how to request your credit hours. And again, thank you all for joining us today. Goodbye.
Bye-bye.
Thank you for your participation. That does conclude today's conference. You may disconnect at this time.